Hi, morning everyone. So as Fran said, my name is Malick. I'm one of the research fellows at OVG. I guess my main passion in life is actually typhoid, but I'm going to spend the next 15-20 minutes just talking about our experience in OVG of conducting some Ebola vaccine trials. But before I talk about what we've been doing in Oxford, I think it's quite important just to contextualise some of the work that we're doing and highlighting some of the what we've learned about Ebola over the course of the past 18 months or so, and talking about some of the leading vaccine candidates. So this cartoon just shows the Ebola virus. So it's quite a long filamentous virus, and as many of you know, if you get infected with this virus, it causes a very severe hemorrhagic fever, and with a mortality rate of anywhere between 50 and 90%. So it's a very severe illness. One of the things that's not really fully understood is if you get infected with the virus and are lucky enough to survive it, whether or not you develop immunity against it. We do know that the virus stimulates a very strong immune response. In particular, this outer protein that's covering the surface of the virus, known as the Ebola glycoprotein, is one of the main targets for the immune response after vaccination. And we know that there are 5 different species of the Ebola virus. They're named after the regions where they were first identified. But the majority of cases historically have been due to the Zaire species of Ebola virus, or Ebola virus, but there are a number of circulating strains out there. We don't really think that there's a huge amount of cross-protection between each of these different strains, so it's something to bear in mind when it comes to designing vaccines. So obviously many of you would be aware of the Ebola outbreak that began right at the end of 2013. And this is just a slide that's taken from The Economist that just gives a bit of context to this. So the virus was actually first discovered in 1976 in what was then Zaire. And then throughout the course of the past quarter of a century, 30 years or so, there have been a number of sporadic cases, typically in Central and Eastern Africa, usually only affecting a relatively small number of people. But as you can see from the slide here, the outbreak that happened between 2014 and 2015 really dwarfed anything that's ever happened before, with over 28,000 cases, and it predominantly affected 3 main countries in West Africa: Guinea, Sierra Leone and Liberia. There are a handful of other cases in some of the surrounding countries, and also interestingly around the same time there was an outbreak in the DRC, which was actually thought to be unrelated to this vaccine that was this outbreak that was going on in West Africa at the moment. So really we've seen more cases of Ebola virus in the course of the past couple of years than have ever been recorded prior to that. So a lot has been learnt about the virus, and the question is really, if we had a vaccine here, could we have prevented this huge outbreak and huge spike of cases? So I'm just going to take a moment just to kind of give you a bit of an overview of how we think the outbreak first started. And I think it's quite important to think about this because it gives you an idea about who you might want to vaccinate if you did have an effective vaccine. So this is Geppetto, which is a region of Guinea, and it is very close to the border of Sierra Leone and Liberia. And this is thought to be the epicenter of the Ebola outbreak. So the first recorded case was a child who was 2 years of age, who had a fever, non-specific illness, became very unwell with GI bleeding and vomiting, and unfortunately died in the beginning of December 2013. Their sister subsequently developed a similar illness, as did the mother and the grandmother of these cases. A nurse who was involved with looking after these patients also developed disease, as did the village midwife, and who passed it on to a family member in another village. One of these healthcare workers visited another hospital in a separate town, subsequently spread to other individuals. And a number of individuals here were infected by attending the funeral of some of these people who originally died. So you can see people who have very close contact with Ebola cases are at risk of developing disease, and also healthcare workers are particularly high risk. And to a certain extent, it's the worst place really for this outbreak to happen. There's a lot of migration between each of these different countries, and this is a road that goes from this region to Conakry, the capital of Guinea. So quite easily the epidemic can spread within this region. And this is a map from the WHO website that shows how rapidly the disease spread, originally starting in this border region. So the blue shows a cumulative number of cases, and the red shows new cases that are reported. So you can see in the middle of 2014 you have a huge explosion of cases all over the region. You have new cases developing in Monrovia, in Freetown and in Conakry, and really almost all of the regions of these countries were affected. Now towards the start of 2015 and over the course of the year, you can see that the number of new cases is diminishing, but there are still sporadic cases popping up in each of the different regions. So this really just highlights how severe this outbreak was and how many people it affected. And thankfully, towards the end of 2015 and the beginning of this year, each of these 3 countries were declared Ebola-free, although if you're following this ongoing story, there have been a few new cases in Sierra Leone after that. So around the height of the outbreak, the WHO realised that the outbreak was getting under control and published this document known as the Ebola Response Roadmap, which included a commitment to try to fast-track access to new treatments and vaccines against the Ebola virus, and try to essentially lay out a framework for how we can accelerate vaccine development, try to coordinate some of the key stakeholders, and also try to put in place a plan to manufacture and stockpile some of the candidate vaccines. So this just shows the epidemic curve in Guinea and Sierra Leone. It shows you have a huge spike in cases and it all falls down here. Now actually the decline in the number of cases was primarily due to effective infection control measures, isolation of index cases, behavioural changes, rather than the introduction of vaccines. So again, we want to think about, could we have done anything before this outbreak if we had an effective vaccine to try to stop it becoming as severe as it was? So even though the number of cases in West Africa has now fallen, the story might not be over. There's increasing awareness that the virus can persist and you can have relapse of disease. This is one case where the Ebola virus was isolated from the ocular fluid of a surviving patient. It can also be isolated from the seminal fluid up until 9 months after vaccination. Many of you will be aware of the Scottish nurse who developed a relapse of disease and was looked after in the Royal Free. And there's also evidence of sexual transmission of the Ebola virus. So this is a patient who survived and subsequently passed it on to another individual who unfortunately died. And I guess the important thing is that this disease, as far as we know, isn't going away. So the animal reservoir for this disease is not fully understood, but it's essentially thought to be have an animal reservoir within fruit bats. So this is a modelling paper that was done by a group in Oxford that overlaid the distribution of 4 different species of fruit bats that are thought to harbour the virus with particular geographic areas where Ebola outbreaks have been described in the past. And in this entire region, there's around 25 million people who are potentially at risk for developing Ebola virus disease. And if you look at this just shows the number of the essentially international travel from the West African region all over the world. So really, as we've seen with other outbreak pathogens in the past, with increased travel, there's always a theoretical risk that these diseases can spread globally. So if we're thinking about developing an Ebola vaccine or deploying an Ebola vaccine, we can either be reactive and try to catch up once an outbreak has already become established, or we can be a bit proactive and develop a vaccine that we have ready to go if this ever were to happen again. And obviously these are slightly different but complementary strategies and may require slight differences in the design of vaccines and vaccine regimes. So, what Ebola vaccines have been developed and what was the history of that? Well, the virus first discovered in 1976, as I said, and really a lot of the Ebola virus research began in the early 2000s, and a lot of this was driven from realization that it could be potentially used as a bioterrorism agent. So a number of vaccines were in early stages of development, typically using viral vectors or DNA-based vaccines. Many of these have undergone testing in some animal models, so there is a non-human primate model and a mouse model of disease, but a limited number of them had undergone phase 1 trials in humans. And then there's a few isolated cases where some of these candidate vaccines were used in laboratory workers who were accidentally exposed to the Ebola virus. But really the message is that none of these vaccines that were in development had really undergone safety testing. just to digress slightly about passive immunisation, so many of you would have heard of the drugs ZMAP or ZMAB, which are experimental therapeutics that were given to Ebola virus patients in high-income countries. And essentially those drugs are monoclonal antibodies that are targeted against that Ebola glycoprotein on the surface, which I showed right at the start. And it's difficult to know if those drugs really work at the moment. But this is a study, this was a report from an outbreak in the DRC in the mid-90s, where essentially they gave blood transfusions from Ebola virus survivors to patients who were suffering from Ebola virus. And just from an observational study that they saw that only one of the 8 people who they gave the blood transfusions to died, compared to the overall case fatality of 80%. Obviously, this wasn't a study that was, this wasn't a formal study that was designed to answer this question, and there's a lot of confounding factors. But around the time of the outbreak in 2014-15, there was a lot of interest in this potential strategy. So a number of trials were set up at that time, including this study in Guinea, which compared giving plasma from Ebola virus survivors to a control. And in this study, they didn't demonstrate any specific benefit from the administration of convalescent plasma, but there's a few other studies that are ongoing, and it's an evolving story. So if we're going to be developing a vaccine against the Ebola virus, ideally we want it to stimulate quite a broad immune response, and a lot of this knowledge comes from animal models. So we know that antibodies and T cells are important in giving protection against disease, and at least in animal models, if you have a particular level of antibody, that gives you 100% protection if you challenge those animals with the Ebola virus. Antibodies aren't the whole story, though. This is a slightly complicated series of graphs to show that T cells are important in giving protection against the virus. Right. So this is one of the leading vaccine candidates, which is known as VSV-ZBOV. So this was originally developed by the Public Health Agency of Canada and subsequently licensed by Merck. And I've got another slide that just gives you an overview of what this vaccine is, but essentially it's a viral vectored vaccine that encodes the glycoprotein of the Ebola virus. And in a number of studies in animals and a few studies in humans, it was shown to be give some protection against the Ebola virus. And one of the main advantages is that it can be given as a single dose. So viral vectored vaccines essentially just follow this platform. So you've got your disease of interest. Could be a virus, could be a bacteria or a parasite. And then essentially what you do is you take a section of its genetic material, DNA or RNA, that codes for whatever antigen of interest. In this case, these red dots that are the Ebola glycoprotein. You then take that bit of genetic material and you transfer it into an unrelated virus that doesn't cause harm in humans. And we call this the vector. So when you put that bit of genetic material inside the vector, that starts expressing the protein of interest, and when you give it as a vaccine, in theory, you make antibodies and stimulate an immune response against it. So there are lots of different vectors that are being used. This slide shows VSV that's used in one of the candidate vaccines. We often use vectors known as adenoviruses and also MVA-based vectors as well. But essentially the principle is very similar. So around the start of 2015, there was the number of cases was falling, but one of these candidate vaccines was at an advanced enough stage to undergo testing in the Ebola outbreak regions, which was the VSV vaccine. So this used a slightly interesting vaccine strategy known as ring vaccination that was originally used in smallpox eradication regimes in the 70s. So essentially what this involves is you have an index case of Ebola, and then you identify the primary ring. So those are the people in immediate contact with that individual, brothers, sisters, relatives, and so on. And then you identify a secondary ring, who are the contacts of those primary contacts. So essentially you define these two rings, and you then randomize each of the groups to receive either the vaccine immediately or to receive a delayed vaccination, so around 3 weeks later. And you then compare the rate of illness in each of those two groups. So obviously, because there's person-to-person transmission, this is one potential way in which you can outline how effective the vaccines are. This is a very long story, and I don't really have time to go through all of this, but these different graphs show the efficacy of this particular vaccine in the study. So the solid line shows the people who received the delayed vaccination. The higher it goes up, the more cases they are. The dotted line shows the people who received immediate vaccination. And as you can immediately see, if you delay giving the vaccine, you have an increasing number of cases compared to if you receive the vaccine immediately. So depending on which way you analyse this data, this vaccine has somewhere in the region of 75 to 100% protection against disease. So it's a very promising vaccine, it seems. There are a few potential issues with it. Some of the early studies shows that you can develop skin rashes or joint swelling after this vaccine is administered. So further work needs to be done about the overall safety of this vaccine. So what we've been doing in the UK, well really the UK, and I suppose Oxford in particular, has arguably been at the forefront of some of these Ebola vaccine development and have benefited from support at all levels of government. And I think one of the most important things is there's been a very flexible regulatory and research environment to conduct these trials quickly, a recognition that there's a need to accelerate vaccine development. So by having frequent and open dialogue between some of the key stakeholders, we've been able to conduct these trials at a very rapid pace. So one of the other candidate vaccines that's undergone testing here is this vaccine ChAd3-ZMAB, which is manufactured by GSK, which uses a adenovirus vector, a chimpanzee adenovirus vector. And in animal studies, it showed 100% protection when given as a single dose. And there was a study that was conducted by our colleagues in the Jenner group testing this vaccine. And broadly speaking, it was shown to be very safe without any major safety concerns and stimulated an increase in your antibody and T-cell response after giving a single dose. So this is undergoing some further testing now, including with booster regimes with other vaccines. At OVG, we've been testing two other leading candidate vaccines, which we called Ad26-ZBOV and MVA-BN Philo. I think we need to work on giving them slightly catchier names. But, essentially, these are using a very similar principle. So these are adenoviral vector vaccines that encode the glycoprotein along the surface of the Ebola virus. And we're testing these really given in combination, where you give a priming dose and then a booster dose after an interval of between 4 to 12 weeks. And in animal studies, these were shown to be very protective. So this is the design of the study, the phase 1 study that we did at OVG, where we had initially 4 groups who were receiving each of these vaccines in different combinations, either 4 or 8 weeks apart. And there was a small group who received a shorter interval between the first and second dose. Some of the people received placebo as well. And there was around 83 people in total. Again, I don't have time to go through all of the results, but broadly speaking, the vaccine was very well tolerated. There was a low rate of fever and a low rate of side effects after receiving the vaccination. But one of the most exciting things really is that the vaccine seemed to be stimulating a very strong immune response. So each of these different panels just represents a different regime, different sequence or different interval between the 2 doses, and the arrow shows the time that they were given. But really, regardless of which regime you give, after giving 2 doses of vaccine, you have a strong increase in your antibody response that persists up until a minimum of 9 months after giving the vaccination. We've just finished the 1-year follow-up for these trial participants. So if we can see persistence of antibody for 1 year or even longer, then that's incredibly promising with these vaccines. Ideally, we want something that gives a durable immune response over a longer period of time. And this shows that you have a similar response when you look at the T cells, which, as I said, we think are important in giving protection against Ebola virus. So this work was done as part of the EBOVAC consortium, and we've moved on to do a much larger study with in a number of different sites in the EU, so primarily here in the UK and in France. And in parallel, there's another study being conducted in a range of African countries that weren't affected by the outbreak to test these vaccines in different regimes and including some of these special groups. And we've just finished recruiting for our part of the study here in the UK. So this is a timeline that shows basically what happened in Oxford really with Ebola vaccine development. So the blue just shows what our colleagues in the Jenner group did. So their trial really first started in the middle of August and vaccinated their first participant on the 17th of September 2014. And after a really rapid period of recruitment, their final participant was vaccinated on the 18th of November. So in between this period of around a month was incredibly busy getting all of the approvals from the MHRA, the ethics committees, and getting all of the contracts signed. And really, our experience in OVG was almost identical. We vaccinated our first participant on the 30th of December 2015, and we enrolled everyone by the middle of February 2015. So, as many people in the audience will attest to, this requires a huge amount of effort and a huge amount of work with everyone pulling in the same direction. And conducting these trials along this timescale is relatively unprecedented, and compared to the conduct of most phase 1 trials is rapidly accelerated. But if I just kind of overlay these dates with the overall Ebola epidemic, then it gives an idea that really, however we can pat ourselves on the back and say we did a really good job getting these vaccines up and running quickly, but really we're always behind the curve. So this is the time when the trial was started in the Jenner group, and this is the time that they had their first participant. Similarly, this was our trial that was started in December and finished in February, and really by that time the number of cases is falling rapidly. None of these vaccines were deployed in the outbreak settings, and this is the time that the ring vaccination study was conducted, so from February to June. So even then, the number of cases seems to be falling down rapidly. So what can we do, or what could we have done, or what can we do in the future to try to have vaccines available at a much earlier stage so that we have vaccines ready to go? It's not all bad news. We've gone from having no proven vaccines to having at least one vaccine with proven efficacy in around 20 months. We've got several vaccines that are progressing through clinical trials here in Oxford and elsewhere. And it's going to be quite important to have several vaccines in development in parallel, primarily to increase the manufacturing capacity in case some of these vaccines don't work or have unanticipated safety concerns. And it may be that different vaccines work better in different circumstances. And this whole process really just kind of highlights how difficult it is really to go from a standing start to try to have an effective vaccine. So one potential strategy that you can use is being more proactive. So the idea is you identify potential outbreak pathogens before they cause epidemics. You preemptively manufacture the candidate vaccines using platforms that are quite adaptable, such as viral vectors. You preemptively do phase 1 trials to establish that they're safe and immunogenic. And all those vaccines that seem to be promising, you stockpile them and you develop trial protocols so that in case of an outbreak, you can rapidly deploy them. This is just an example of an outbreak of Lassa fever that started in August 2015, was ongoing in January of this year, that affected 159 people in northern Nigeria with 82 deaths. And this is a disease that's not too dissimilar from Ebola virus. So essentially, these outbreak pathogens are likely to rear up again, and we need to be better prepared. And partly as a result of that, the UK government has promised investment of, I think, around £130 million to try to accelerate research for these outbreak pathogens so that we're better prepared if anything like this were to happen again. So many vaccines have undergone safety testing. Several of them have progressed to phase 2 and 3 trials. Despite some significant achievements, the response ultimately has probably been too slow, but I think the field has changed dramatically over the course of the past couple of years or so, so hopefully next time we'll be much better prepared. None of these studies in Oxford and elsewhere would have taken place without everyone pulling together. There's a huge number of people to thank who actually contributed to the delivery of the studies, and we've obviously been working with partners both here and abroad. So that's it. So thanks.